A warm good morning to one and all. We are continuing with vectors. Previously, we had discussed about the plasmid vectors, and today we will finish the remaining type of vectors which can be used in genetic engineering. If you go to consider a bacteriophage, we know that it can do both a lytic cycle and a lysogenic cycle. What is a bacteriophage? A phage is something which will infect a bacteria and it transfers its DNA into the bacteriophage. And if the bacteriophage is undergoing a lytic cycle, you can see that the lysis of the cell will happen when the macrophages are being released. And you also have another stage called the lysogenic phase where the phage DNA is remaining integrated with the bacterial DNA for a period of time. Whatever be the case, we can see that when the phage DNA enters into the E. coli, phage DNA as such, when it has been present in the phage, it has been found to be linear. But once it enters into the bacteria, it circularizes and forms a circular form of DNA. And under the influence of the E. coli DNA, multiple copies of the phage is being created in the lytic cycle, and as well as different parts of a phage is being formed and they are being assembled. And once the assembled phages are being formed, the cell lyses and the phages are being released out. This happens in the lytic cycle, but in the lysogenic cycle, the DNA of the phage gets integrated with the host, that is E. coli here, and it forms a prophage stage. And then multiple copies of the phage is also being created, phage DNA is also being created, and in between, what happens? The prophage may leave the bacterial chromosome also. So this is a lysogenic stage as well as you have the lytic stage. So this is what we have to say about the phages. Now, if you go to look into the phages, the phage DNA as such is consisting of a lots of, I'm talking about the phage DNA. It has a contamer molecule when the DNA synthesis is being carried out in the E. coli. Now, what is a contamer molecule? It is a long continuous DNA molecule that contains multiple copies of the same sequences linked in a series. And usually, these polymerase molecules are usually copies of the entire genome linked end to end and separated by cos sites. Cos sites are cohesive sites and a protein binding nucleotide sequence that occurs in each copy of the genome. So when the phage DNA is being multiplied within the E. coli, they are usually formed as contamer molecules. That is, the entire genome of the phage is repeated and they have been formed as different chains. And at the end of each genome, there will be present certain cohesive sites or the cos sites. Now, what are these cos sites? Cos sites means they will be having sticky ends. That is this end and this end. If you go to cut it into each genome, if you consider this genome, what happens? This end and this end will be sticking, or it can be able to, they will be overlapping with each other. So when a bacteriophage is being formed, every genome or every part, every segment of the cos site containing genome, okay, so the cos site has been formed only at the ends of the genome, will happen. So this entire, this what is it, segment containing two cos sites will, that is every fragment of this bit which has been cut over here will enter into each virion and it will form a complete virion. So if we go to consider when a bacteriophage is attaching the thing, it has a DNA and this is the genome. This is the feature of the genome which happens to enter the E. coli. It is having cohesive ends or the cos sites which will be here. And once it enters into the E. coli, it will circularize and sometimes it might also get integrated with the E. coli genome and form a prophage. So this is what normally happens within a phage DNA during its replication. And you can see that if you have a different contomer with containing multiple cos sequences as well as genome, only one of that enters into the head. That is it. If you go to look at the DNA, this round ball thing is a cos sequence. So the phage DNA, you will have two ends to cos sequences, and it will multiply. Now, what does this have to do with a cloning vector? Now, if you go to consider the phage DNA, you can see that the 60% of its genome is needed for its lytic cycle, and there are certain non-essential regions in the phage genome. So, we have utilized the phages as a good vehicle for recombinant DNA technology by removing these non-essential regions. So, which regions are non-essentials? So, that region is being removed from it, and here we can put a foreign DNA can be inserted. So, if you remove this non-essential portion, you can use this phage DNA as a vector for recombinant DNA technology. And the advantage of this is that, unlike about approximately 10 kb we put in plasmid vectors, in the case of phage vectors, you can put approximately 23 kb of the foreign DNA can be put into a phage cloning vector. So, as I told you earlier, the DNA of the phage is in the form of linear molecule which gets circularized once it enters into the E. coli. And at each end, there are short single-stranded 5' projections of 12 nucleotides, which is complementary in sequence. And these DNA adopts a circular structure when it is injected into the host cell. And the lambda DNA naturally has a cohesive termini, which can associate it to form a cos site. Now, if you go to look into the lambda vector, the lambda vector can incorporate large amount of DNA, and hence we can use lambda vectors for the construction of genomic libraries. To package these bacteriophage lambda DNAs into it, the what you have to do is you need two cos sites at one at each end, and usually 50 kb apart. That is, these two cos sites are being kept 50 kb apart, and this ensures only 50 kb gets, that is only 50 kb gets enters into the head of it. So if you remove the remaining portion of, or the unwanted portion of the DNA from the lambda phage, you can use it as a vehicle. So consider this as to be as a what phage DNA. It has some sites for echo R1 and all that. And what you do is you can cut it with echo R1 and remove all the unnecessary stuffers or the non-essential segments of it, and you can add your what essential DNA, that is your vector, the foreign DNA with which you have to incorporate, and you can put it into it. So the phage DNA as well as your own DNA which were needed to be incorporated into the vector will be cut with echo R1, and that is put into it. You will get a recombinant DNA. Now, once this recombinant DNA, so as I told you, the unessential elements are removed and the vector needed one has been put into it, you get a recombinant DNA. And that, and along with the phage packaging system, which we will artificially create in the recombinant DNA technology. And so what happens? They will become an infectious phage. And the infectious phage now can infect the normal bacterial cells, and they will form plaques. You know what is a plaque? A plaque is when the bacteriophage grows onto the E. coli cell, it will form some clearance zones. And you can identify if the bacteria has grown, bacteriophages invaded or grown on the bacteria here. And one some what such type of phage is a lambda phage. You have Charon 4A lambda. These are all examples of some vectors which are being used, some phage vectors. One feature of these phage vectors is that they also have the galactosidase system, as we had mentioned in laxi system of plasmids. And with xgal, they will be producing beta galactosidase, and the colonies will be turned into blue. So when a DNA segment replaces the stuffer region, the LAC5G is missing in the case of phage vectors, and as a result, what happens? Beta galactosidase is not being produced, and all those colonies which contain the what which will contain what? Consider thing, the recombinant DNA will not contain the beta galactosidase, and as a result, what happens on the plate when they are being forming here? See, in this plate when they form, they will be forming white colonies if they contain the recombinant DNA. And if the recombinant DNA is not being present, and if only the phage vector is being present, blue colonies will be formed. So the screening of these phage vectors is being carried out by the process of what? Blue-white screening, as we had mentioned in plasmid, in the case of plasmid itself. And here you can identify the recombinant cells with the presence of white colonies. Now, they have some advantages over the plasmid system. You can have insert a large amount of DNA of approximately as 23 kb, and the packaged phage is usually highly infectious and infect E. coli at a higher efficiency than the plasmid transformation methods. So it's an easy method to infect the host cell or the E. coli cell over here. And you can see that here the positive clones or the plaques which can be obtained here can also be further screened by other techniques by called the plaque hybridization techniques. Now, if you have multiple colonies, you can also use the technique called the plaque hybridization. What is plaque hybridization? That is, once you have a plate having agar plate having plaques, you can take and keep a filter paper over here, and onto the filter, what happens? The DNA of these plaques can be filtered. Like, there are some treatments which you do, I'm not going to the details of it. So you do some procedures so that these cells get lysed, and the DNA of these plaques will happen to get filtered onto this filter paper. And you can use this filter paper later on by various other techniques called autoradiography, by using labeled probes to find out if specific DNA is being present on this. So in the case of phages, we can identify the recombinants by blue-white screening, as we had mentioned earlier. Blue colonies will be used to identify the non-recombinant ones, and the recombinant DNA will be present in white colonies. Further on, if you want to find out the presence of specific DNA on a colony, you can also use techniques such as a plaque hybridization, where the plaques have been transferred onto a filter paper. Specifically speaking, the DNA of the plaques have been transferred onto the filter paper, and then you will take this filter paper, and all the non-specific DNA and protein are being removed by certain steps, and you hybridize it with a labeled probe and look it and detect it by autoradiography. So where the specific DNA is being formed, you will form a black type of marking, indicating that your DNA in all the different clones, it is at this particular region that the specific DNA is being present over here. So I guess you got an idea about phages and how they work as a vectors and how they can be determined. Thank you for now.